replication. So DNA replication. So for DNA replication, the objectives that we need to achieve by the end of the study are that we are supposed to be able to cite evidence that two scientists did. So there was a scientist named Michelson, and another one was the scientist Stoll. So they did an experiment that enabled scientists to come up with or to differentiate between conservative DNA replication and other alternative models of DNA replication. So during the study of DNA replication, the scientists needed to prove which model, which hypothesis does DNA replication follow. Is it a semi-conservative or it is another alternative model? So there was an experiment that was carried out by these two scientists. So we need to understand that experiment. And then we need to summarize how DNA replicates and also to be able to identify some features that are unique for this process. Then it is important that we know the proteins. We need to know the enzymes that are involved in DNA replication. We need to know the complexities that are associated with DNA replication, which makes it to be bidirectional. So which means that DNA replication moves in two directions, and also that it is continuous in one strand and discontinuous in another strand. And then we need to understand how the leading and the lagging strand happens, the synthesis of these two strands. And then also need to know how enzymes are able to proofread and to repair errors that happens during DNA replication. And then finally, we need to know how does DNA replication end? What is the termination? How does it terminate? So these are some of the objectives. If these objectives are met, then DNA replication will be easier for you, to be easier for us. So almost any question will be able to answer any questions if we are able to understand these objectives. So we are going to talk about the three alternative models of DNA replication. And then we are going to talk about this experiment, the Michelson-Stoll experiment. Remember, the Michelson-Stoll experiment needed to prove which model of DNA replication is correct. And then we're going to look at the mechanism of DNA replication and also how replication terminates. So this is just a sketch outline of what we are going to look at. So these are the three alternative hypotheses. The three alternative hypotheses. So these are the three alternative models, views of how scientists thought DNA replication happened. So the first one, this one, on your left, this one is what is called the conservative model or conservative replication of DNA. So this, the blue color represents the parental DNA strand. So remember, DNA is double-stranded. So you have these two strands. You have the other strand. So the blue strand represents parental strands, or the strands before DNA replicates. And then the golden color, this one, the brownish color, represents the daughter strands or new strands. So when we look at the first hypothesis, the first hypothesis is called conservative theory or conservative hypothesis of DNA replication. So when we look at this one, the conservative replication simply means that if these are bases in the parental DNA strand, these bases in the parental DNA strands are turned outwards so that we have complementary strands that no longer face each other. These could be used as templates. This could be used as a template for the synthesis of an entire new double helix all at once. So what they are saying is this. Remember, DNA double-stranded. So each of these strands can be used to synthesize an entire new strand. So remember the orange, this color, the golden color represents new strand. So under conservative, this hypothesis is called conservative because this, after replication of DNA by this method, the hypothesis is that this would result in an intact parental molecule. So you have an intact parental molecule. Remember, the blue represent intact, represent parental molecule. So DNA replication by conservative hypothesis simply means that after DNA replicates, this will result in an intact parental molecule. So this parental molecule is still intact, still double-stranded DNA containing those two strands from the parent. And also a daughter molecule, a daughter DNA molecule consisting entirely of newly synthesized strands. So remember, conservative form of DNA replication says that DNA replication will result in an intact parental molecule, and a daughter DNA molecule consisting entirely of new synthesized strands. So this is conservative DNA replication. And then we have semi-conservative DNA replication, semi-conservative DNA replication. So when we talk about semi-conservative, so it means that if this odd strand, this odd or parental strand of DNA is separated, and then each strand acts as a template for the synthesis of a new daughter cell. So each of these two strands will act as a template for the synthesis of a new daughter strand. So we have the odd strand, this odd parental strand, acting as a template to synthesize a new strand. Then the other parental strand will also be used to synthesize another new daughter strand. So by semi-conservative, it is called semi-conservative because each new daughter DNA molecule. So remember, we have two DNA molecules, this molecule and this one. So each daughter new DNA molecule would consist of one odd strand, the blue one, and one new strand. So one odd strand and one new strand. So this is semi-conservative. And then we have the dispensive model of replication. So you have the double-stranded parental molecule. Okay, you have a double-stranded DNA molecule. If this parental double helix okay, so you can unmute when you want to speak. I was hearing some background noise. Yeah, so we have this double stranded parental molecule. If this parental double helix is cut wherever one strand crossed over another, and then DNA is synthesized in short sections that extend each of the cut parental strands to the next strand crossover. Then there would be a mixture of new, so you have the new and the odd. New and odd segments along each replicated molecule. So under this dispensive hypothesis, it is stated that after DNA replication, then each new daughter strand would contain a new segment, which is that golden color, and an odd segment. So it is going to contain new segments odd segments, new segments, odd segments. So it contains both segments in each strand. So this is what is called the dispersive replication. So we have three alternative hypotheses: conservative, semi-conservative, and dispersive. So now for the scientists to prove which hypothesis was correct, what they did, two scientists did an experiment. They did an experiment, which is called the Michelson-Stowe experiment. So these three models of replication that we've looked at were evaluated by Michelson and Franklin Stowe. So for them to distinguish between these three alternative models of DNA replication, they labeled DNA and then followed the labeled DNA through rounds of replication. So what they did, of importance, they used the bacterium E. coli or Escherichia E. coli. Escherichia coli. So they used the bacterium E. coli. E. coli is a bacterium that they used. So E. coli is very advantageous for them to have used it because it is small and is able to grow quickly and readily in the lab. So no wonder it was a favored bacterium to be used for this experiment. So what they did is that they grew this bacterium on a medium that contained heavy nitrogen. So heavy nitrogen is 15N, the 15N. So this is heavy nitrogen. So 15N. So they grew this E. coli in 15N in the form of ammonium chloride. So that is 15N. So you understand that 15N molecules or molecules that contain 15N have a greater density than those that contain the common 14N, the 14N or the light nitrogen. So 15N is more dense than the 14N. So after that, the other thing that you need to note is that they used density gradient centrifugation. Density gradient centrifugation. So what they did is that the DNA was mixed with a solution that contained calcium chloride. So calcium chloride, which is CSCl solution. And then they centrifuge at high speed. So I find that when that is done, the solution is going to form a density gradient in the centrifuge tube, which will range from a region of lowest density at the top. So at the top, you have low density at the top, and then to one of the highest density at the bottom. So remember, the more dense a substance, the lower it is going to be in the tube. So the more dense will be at the bottom, the lightest will be at the top. So we find that during centrifugation, the DNA molecules migrate to the region of the gradient that is identical to their own density. So this is a summary of the experiment that they did. So they grew E. coli in heavy nitrogen for many generations, so that those DNA strands would be heavy. So the DNA strands would be heavy. And then they transferred some of the cells to 15N or light nitrogen medium, so that those new synthesized strands would be light. So they would be less dense than the 15N. And then they isolated those DNA from the bacterial cells after 20 minutes. So this 20 minutes is a generation for the E. coli, and also after 40 minutes, which represents 2 generations. And they did the centrifugation, as I have explained earlier, in order to separate this DNA into their bands, depending on how heavy they were. So when they put when they centrifuged the DNA from the 15N, after growing it in the 15N, before they transferred it to the 14N, what happened that all the DNA was heavy. So it was 15 DNA, 15N DNA, so it was heavy. So it was at the bottom. So this is very important. In 15N, everything, the DNA was heavy. It contained heavy DNA. So it was heavy DNA. So the DNA collected immediately here was 15N. It was heavy. After that, remember they transferred it to 14N. So in 14N, what happened that after one cell generation, after 20 minutes, what happened that a hybrid DNA was produced. So the result was an intermediate. It was an intermediate. It was intermediate. So it contained both heavy DNA and light DNA. So it contained heavy DNA and light DNA. So no wonder you find that in the centrifuge tube, it was intermediate. It was in the intermediate. So it contained both heavy and light. After one generation in 14N, it was intermediate, heavy and light, a hybrid DNA, very important. And then after 2 generations in 14N, what happened is it was a mixture of light DNA and a hybrid DNA. So light DNA and a hybrid DNA. So that's important. So this is it. This is a semi-conservative hypothesis. So after 2 generations, which is here. Remember I said that after 2 generations, there was 14N light DNA and a hybrid DNA. So that was 50%. So half were low density or light, so containing 14N, and half were intermediate density. So it was a hybrid DNA that was produced after 2 generations in 14N. So remember this. Remember that in 15N, before it was transferred to 14N, it contained only heavy DNA or 15N DNA. Then after 1 generation, 14N, it contained a hybrid. So it means that it had 14N, 15N, hybrid DNA. And then after it had stayed for 2 generations, in 14N, what was produced was 50% 14N DNA or light DNA and 50% 14N, 15N hybrid DNA. So what this experiment proved was that actually DNA was semi-conservative, as we can see here. Because the result of after 1 generation, after generation 1 in 14N, was an intermediate or a hybrid, which agrees with the same conservative DNA replication. And then after generation 2, we find half low density and half intermediate. So this is what the experiment of Meselson-Stowe experiment proved. So it proved that DNA actually replicates by semi-conservative. So DNA replicates by semi-conservative. So this experiment led to the conclusion that each newly made DNA molecule comprises of each newly synthesized DNA molecule. So the DNA double-stranded. So each newly synthesized DNA molecule or double-strand DNA comprises of 1 odd, the blue one odd strand and 1 new strand. Therefore, DNA replicates by semi-conservative. Okay. So now, having looked at this, we need to understand how does DNA replication happen? What's the mechanism of DNA replication. So we need to understand, first of all, that DNA replication is bidirectional and semi-conservative. Remember that we've explained what semi-conservative means. Said that after replication, it means that each daughter molecule of DNA will contain one intact parental strand and one newly synthesized strand that will be joined by base pairs. So remember, one strand of DNA is adjoined to the other strand of DNA by base pairs. So remember, guanosine will form hydrogen bonds with cytosine, 3 hydrogen bonds. And then we have adenine and thymine forming hydrogen bonds, 2 hydrogen bonds. So adenine on one strand and thymine on another strand forming those bonds. So DNA is semi-conservative. Then the other one is that DNA is bidirectional. So it means that DNA happens or moves out in 2 directions. So there's what is called the replication fork, and then we have the site at which a replication begins, the origin of a replication. So from the origin of a replication, DNA will move in both directions. DNA will move in both directions. This is what it means by bidirectional. So therefore, replication bubble grows in 2 directions as DNA replication proceeds. So we have unicellular organisms, which are the prokaryotes, and multicellular organisms, which we call the eukaryotes. We are going to look at these cells in an individual lesson. We are going to look at the prokaryotic cells and eukaryotic cells. However, we find that in prokaryotic cells or organisms, these have only one site of origin of DNA replication. However, for eukaryotes, there are multiple sites of replication. And of interest for us is DNA replication that happens in prokaryotes. Of course, we are going to add a certain concepts for eukaryotes where it is important. So DNA is bidirectional and semi-conservative. Is it clear so far?
Sorry, I was logged out.
Sorry, speak up.
I was logged out. When you just introduce that this part. This is when I was logged.
Okay, where did you log out from?
Notification is bidirectional and conservative.
Okay, so this is where we still are. So any questions so far?
No question.
Okay, so bidirection simply means from the site of origin of replication, DNA replication moves in both directions. So it is going to move in 2 directions. One forward, one backward. One forward, one in this direction, in the direction, and the other one will be moving in the other direction. So we're going to understand when we talk about the lagging strand and the leading strand synthesis. Then semi-conservative, we said that each daughter cells after replication will contain one parental strand and one newly synthesized strand. And then we have replication forks. So these are simply the Y-shaped region where the parent DNA strand split into 2 single strands, and henceforth those strands are coupled. So replication forks are the sites at which DNA synthesis is occurring. So I'm going to show a diagram later, which will show the replication fork, and now in a diagram, the grammatic view of how DNA replication happens. So we have the replication fork, which is a site at which DNA synthesis will be occurring. And then you need to know that there are specific set of proteins that are responsible for recognizing these sites where replication begins and opening the double helix at those points. So these proteins are activated by the proteins that initiate S phase in the cell cycle. So we're going to understand more on the cell cycle when we start looking at the genetics. on just looking at cell division. However, we have proteins that are able to recognize these sites for DNA replication to begin. So we have proteins such as DNA A proteins that are able to initiate this process. And then DNA of importance also is that DNA replication, and as we are going to see further, transcription as well. The initiation typically begins at adenine and thymine base pairs, sites which are rich in adenine and thymine are base pairs. So remember, adenine and thymine, as I explained earlier, these are paired with a double hydrogen bond. And then guanine and cytosine are paired with a triple hydrogen bond. So I find that these that are paired with a double hydrogen bond, they are very easy to separate. And it will require relatively less energy to break these double bonds. So therefore, it is easier to unwind DNA or to separate DNA molecule at the adenine thymine base rich sites. So DNA replication begins at adenine thymine base pairs rich sites. So you need to note that. And then we have the enzyme DNA polymerase. So DNA polymerase are the enzymes that are involved in the catalyzing of the synthesis of DNA. Now, the complexities of DNA replication comes about because of how the DNA polymerases function. So the DNA polymerases can only copy a DNA template in the 3' to 5' direction. And henceforth, it is going to produce newly synthesized strand in the 5' to 3' direction. Remember, DNA molecule is double-stranded, and those two strands are antiparallel. So which means that they move in two different directions. So one strand is moving in the 3' to 5' direction, and the other strand is moving in the 5' to 3' direction. However, when you look at DNA polymerase, it only copies a DNA template in the 3' to 5' direction. So this poses a complexity and leads to us having what is called the lagging strand and the leading strand, as we are going to see further. So this DNA polymerase enzyme is able to add deoxyribonucleoside triphosphates, which are the precursors for DNA synthesis. So remember, DNA is a polymer of nucleotides. So the DNA polymerase is able to add nucleotides. So it adds nucleotides. So each precursor pairs with the corresponding base on the template strand, which leads to a formation of a phosphodiester bond with the hydroxyl group on the 3' of the sugar at the end of the growing chain. So a phosphodiester bond forms at the 3' end. 3' end carbon of the sugar. And then DNA polymerase cannot initiate a DNA replication. So it needs what is called a primer. So it needs an RNA primer, which is simply a strand of few nucleotides that is bonded to the template strand. This provides DNA polymerase with a free 3' hydroxyl group, which can be used to combine with the incoming nucleotide, deoxyribonucleotide, to form that phosphodiester bond that we talked about at the 3' end. So it needs a primer. So it's very important to understand that DNA polymerase cannot initiate DNA replication. It needs a primer or primers. So we're going to see that further, we're going to see further that DNA in the lagging strand, DNA replication in the lagging strand will require multiple primers. So to need numerous primers, to need many primers, but in the leading strand, it will just need a single prime. So as I've already mentioned that it cannot initiate synthesis of new strand. So it works only in the 5' to 3' direction. Remember, it adds the nucleotides at the 3' end. So to work in that direction. So it requires both a 3' end to extend from and a single-stranded template. So no wonder the double-stranded DNA molecule has to be split so that each of those DNA strands are used as a template. So I've already mentioned that the primer is an RNA primer. So RNA will serve as a primer. So the bonding of the primer is characterized by an enzyme called primase. So very important, primase characterizes the synthesis of the RNA primer. So primase enzyme, it's an enzyme. So this is an RNA primer. So primase is going to synthesize this short segment of RNA. So this is 3' to 5' direction. This is a template DNA. Remember, polymerase will add at the 3' end. So to work or synthesize in this direction, it is going to elongate the new synthesized DNA by adding a deoxyribonucleotides at the 3' end, provided by this hydroxyl group there. Makes it possible for the phosphodiester bonds to form. So this is the RNA primer. So primase will synthesize this RNA primer. Okay, then we've already mentioned that then the DNA polymerase will add the nucleotides at the 3' hydroxyl of the RNA primer, and then subsequently to the ends of the growing DNA strand. So to just continue adding at the 3' end. And then, remember we said that DNA replication is bidirectional. So it means that these DNA parental strands are copied simultaneously at the replication fork, though they are going to run in opposite directions. So it's bidirectional. Okay, remember we said that DNA is described as being antiparallel. So one strand runs in the 3' to 5' direction, while the other strand, which is complementary, will be running in the 5' to 3' direction. So this nature of DNA, combined with the nature of how polymerase enzyme works, makes this process of DNA replication so complex. Remember that the DNA polymerase can only synthesize only in one direction. However, the two DNA strands run in opposite direction. Therefore, it means that also DNA polymerase is going to synthesize DNA in opposite direction. So DNA or DNA polymerase will require multiple primers on one strand, and it will only require an initial primer in another strand. The one which requires just one strand is what is, the one which requires just one initial primer is what is called the leading strand. And this strand is synthesized continuously. Since it only requires one primer, there will be no gaps. However, when we look at the lagging strand, since it needs multiple primers, it means that it is going to be copied discontinuously. So there will be more like gaps between segments. There will be gaps between segments. This is what is called the lagging strand. So leading strand requires one strand of prime, or one initial prime. The lagging strand will require multiple primers. Now, those short DNA segments or fragments that are synthesized on the lagging strand are what are called Okazaki fragments. So they are called Okazaki fragments. Remember, lagging strand, you have the primer, then DNA adds deoxyribonucleotides, and then you have a gap, you have another primer, and DNA polymerase adds nucleotides, and then you have a gap, and then you have another primer, just like that. So those DNA fragments are what are called Okazaki fragments, Okazaki fragment. So of importance that the leading strand is synthesized continuously in the strand that runs in the 3' to 5'. Remember that the newly synthesized DNA will run in the direction 5' to 3' direction, because DNA polymerase will be adding at the 3' end of the newly synthesized DNA. So the leading strand is synthesized in the parental strand that runs from 3' to 5'. And this is done towards the replication fork. So it's done towards the replication fork. So this is the leading strand or continuous strand, because it leads into the replication fork and it is continuous. It does not have any gaps. Then this lagging strand is synthesized in a discontinuous manner. And this is synthesized in the parental strand, which runs from the 3' to 5' away from the replication fork. So away from the replication fork, which means that the newly synthesized DNA strand, or the newly synthesized DNA strand, yes, will be in the 5' to 3'. So it needs to be complementary to the parental DNA strand. So this is the lagging strand or discontinuous strand, because it lags behind the synthesis that is occurring at the fork, replication fork. So know that the leading strand is formed in the by copying the parental strand that runs in the 3' to 5' direction toward. So the leading strand is toward the replication fork. The lagging strand is away from the replication fork and copied in the parental strand, which runs from the 3' to 5' direction. Okay. So just as I've already mentioned, DNA polymerase will move along in the 5' to 3' direction. And then it is going to be, this is in the lagging strand. Those primers, since they are RNA primers, what we need are DNA bases, DNA molecules. So what will happen is that RNA polymerase 1 will move along in the 5' to 3' direction, removing those RNA primers ahead of it. And then it is going to replace these RNA nucleotides or ribonucleotides with appropriate deoxyribonucleotides. And then once these RNA polymers are removed and replaced by DNA, we have an enzyme called ligase, DNA ligase, which is now going to catalyze the formation of phosphodiester bonds to form between these adjacent fragments. So what we are saying is this: DNA polymerase 1 will move in the 5' to 3' direction after there's a synthesis here, and then it is going to be removing these RNA primers and replacing these with appropriate deoxyribonucleotides. So for example, in a DNA molecule, you're not supposed to find uracil, you're supposed to find thymine. So it is going to replace this uracil base with a thymine base. So there you're going to have T. And then there you're also going to have T. And then after that, we have DNA ligase, which is going to join these segments. So now synthesis of the lagging strand. Lagging strand synthesis, it's very easy and straightforward. And we've explained much of how that happens. So remember, DNA is opened and unwound, and then we have one primer. So primase, RNA primase will synthesize a primer. Synthesize a primer. So one single primer for leading strand, one single primer for leading strand. And then remember, RNA then will start adding deoxyribonucleotides at the 3' hydroxyls, henceforth extending this new synthesized DNA strand in the direction or towards the replication fork. Remember, the replication fork is the Y shaped. So it will be synthesizing the DNA strand towards the replication fork. So DNA A proteins do the initial recognition of the site of replication, or the site of origin of replication. So DNA protein does that. And then we have the enzyme helicase. The enzyme helicase opens the double helix at DNA molecule. Now, when that double helix DNA is opened, this destabilizes the DNA molecule. So what will happen is that we have a single strand DNA binding proteins that are going to stabilize the single strands. So these are single stranded DNA proteins will prevent the reformation of the double stranded DNA helix. So they stabilize the single strands. Helicase opens the double strand. Single stranded binding proteins stabilize the single strands. So we need to know this. Then topoisomerase enzyme has to relieve twisting forces. So the topoisomerase prevents excess coiling of the DNA strands during replication. Then we have the synthesis of the lagging strand. Synthesis of the lagging strand. So lagging strand synthesis. So remember, the lagging strand synthesis needs multiple primers. So for example, we have one primer here. We have the single binding proteins. We've explained what they do. Helicase, we have topoisomerase doing their functions. These are still single-stranded binding proteins. So we have the primer that is added by primase to initiate DNA replication. And then DNA polymerase 3 now will work in the 5' to 3' direction, synthesizing the first Ogasaki segment or fragment on the lagging strand. So this is the Ogasaki segment. Ogasaki segment. So what will happen then? We also need a second primer. So a second primer will be bonded there. And then DNA polymerase 3 will synthesize the second fragment, Ogasaki fragment. And then these primers are continuously being added. They will continuously be added. Then remember we mentioned that DNA polymerase then will start moving along the 5' to 3' direction. So from here to be moving in this direction. There it is going to remove the RNA primers. So to be removing the RNA primers and then replace those with the deoxyribonucleotides. And then remember, after that we have DNA ligase. So DNA ligase is going to close the gap in the sugar phosphate backbone. So DNA ligase will close these gaps that were created, these gaps, it's going to close the gaps. These gaps will be closed. It has to close these gaps. Henceforth, you have a continuous strand there because those gaps have been closed up. So this diagram summarizes or puts everything that happens during DNA replication in one diagram. Remember, we talked about a helicase. We talked about helicase. So this is the helicase, which does the unwinding of DNA. The unwinding of DNA. So remember, the DNA proteins are going to attach at the origin of replication. That results in the formation of the initiation complex. So remember those proteins that we said recognize the sites. So DNA proteins are going to attach at the origin of replication, which is going to result in the initiation complex. And then we have DNA denaturization, which is simply the unwinding of the DNA molecule. This is going to be done by the enzyme helicase. So helicase does the unwinding. So this is helicase, this is a helicase. And then we have the single binding proteins, single-stranded binding proteins, single-stranded binding proteins on both the leading, the lagging strand and the leading strand. So remember, the leading strand is towards the replication fork. So this is the replication fork. Then we have topoisomerase which relieves coiling or tension or torsion. So it prevents supercoiling of the DNA molecule. So we have the RNA primer, we have the Ogasaki fragment growing in this direction. We have RNA ligase joining these gaps or filling up these gaps. RNA primer being replaced by DNA nucleotides by DNA polymerase. We have the DNA polymerase which synthesizes DNA. So this is a diagram which simply summarizes everything that we've talked about. So this is still DNA replication diagram showing how synthesis happens. Just an overview or a summary of how DNA replication takes place. And then after DNA replication has happened, we understand that the fidelity or the ability to accurately replicate a DNA is actually very high. However, we find that in some cases there could be errors. So what will happen is that there is a process of proofreading that happens. So DNA has to be proofread. So there's the proofreading function. So errors, for example, there could be an insertion of an appropriate nucleotide during the process of replication. So what will happen that by the exonuclease activities of the polymerase enzymes, these are going to be replaced. So for example, if where there is supposed to be thymine, a wrong base of uracil is replaced or is placed there, or guanosine bonding with adenine, that need to be replaced by an appropriate base. So that is a proofreading function. Then it could also be the repairing process that can be done. So there are also post-replication repair processes. So DNA can undergo repair. So if for example, there's a segment that contains a damaged region, so that segment can be removed. And then there could also be filling in that gap. So DNA polymerase is able to fill in that gap where that segment has been removed. And then ligation or the joining of the newly synthesized segment to the remainder of the chain has to happen. So there's that fidelity. Now, termination of DNA. DNA has to, DNA replication has to terminate. So we find that in prokaryotes, it will occur at corresponding points of the genome, which is the termination site. So two factors are going to determine this. So you have the meeting replication forks. So the replication forks are going to meet there will be convergence of two replication forks meeting. And then there are also what we call DNA replication terminus binding sites, or the TAR sites. So the meeting of the forks will occur that DNA molecule as a closed circular shape. So most of the prokaryotes of bacteria, for example, have a closed circular shape. So when those two replication forks meet, as a result of that continuous synthesis, the 3' and the 5' ends of each chain are going to meet and join, which is going to lead to DNA termination. Remember the structures which are called the catenins. These are simply topologically linked circular molecules. So remember, after DNA replication in these prokaryotes, they are going to be topologically linked. They are circular molecules, so they are going to be more like adjoined to each other. They'll be linked to each other. So these need to be broken down. So DNA gyrase. So this is a topoisomerase. This is a topoisomerase. This DNA gyrase, topoisomerase in prokaryotes, it's going to separate these rings, and then the process is completed. Then we also have the terminus utilization substance. So the TAR sites are also involved in termination. So they contain a short sequence to which this protein product of the TAS, or terminus utilization substance, gene binds. So this gene encodes the DNA binding protein TAS that, when bound to the terminator site, is going to block a replication fork. So this TAS is simply a protein which binds to terminator sequences, and hence it's going to act as a counter helix when it comes in contact with an advancing helicase. Remember, helicase is one which is unwinding or opening up the DNA molecule. So the moment the helicase meets this TAS, a protein, it is going to block the further advancement of the replication fork. Henceforth, it is able to terminate. Then remember, termination of DNA in eukaryotes. So we find that one chromosome is going to contain several points of replication initiation, and therefore termination can occur in two cases. It is going to occur when there's a collision or convergence of replication forks that are moving in opposite directions. And then the other one is when the replication reaches the end of the chromosome. So at the end of the process, the separated DNA molecule are going to bind to proteins, chromosomal proteins, and then distributed in an orderly manner among those daughter cells. Now, what happens? What are the events really that happen during termination? So termination has occurred or occurs when two replication forks meet on the same stretch of DNA. So what will happen is that those forks are going to converge until all the intervening DNA is unwound. And then remember, there are gaps in DNA. Those gaps have to be filled and ligated. So have to be filled and then joined, which means that now DNA synthesis has completed. And then the catenins that we've explained earlier, that these are topologically intertwined molecules or strands. These catenins have to be removed by DNA gyrase or type 2 topoisomerase. So the catenins have to be removed by type 2 topoisomerase. Then remember, we have the replication proteins. Those have to be disassembled or they have to be unloaded. The replication proteins have to be disassembled. The helicases, single-stranded binding proteins have to be unloaded. So those are the replication proteins. The DNA polymerase have to be unloaded. So lastly, what you need to know are the functions also of the different proteins and enzymes. So you need to know what's the function of helicase. So helicase is going to catalyze breaking of hydrogen bonds between base pairs to open the double helix. So helicase is responsible for the opening or unwinding of the double-stranded DNA. Remember the single-stranded DNA binding proteins, these are going to stabilize single-stranded DNA, stabilization of single-stranded DNA. Remember we have the primase. So primase on the leading strand is going to catalyze the synthesis of the RNA primer. Remember the leading strand only has one RNA primer. And then primase on the lagging strand is going to synthesize RNA primers on the lagging strand. So primase catalyzes the synthesis of the RNA primer on an Okazaki fragment. Then we have topoisomerase. So remember, topoisomerase prevent the supercoiling of the DNA molecule. So topoisomerase prevents supercoiling of the DNA molecule. And then you have DNA polymerase 3, which is going to act to extend the leading strand and also extending the Okazaki fragments in the lagging strand. Remember, DNA polymerase 1 is one which removes the RNA primer and replaces that with the DNA nucleotides. And then we have DNA ligase, which is going to catalyze the joining of Okazaki fragments into a continuous strand, Then we have the sliding clamp. The sliding clamp simply holds the DNA polymerase in place as it is moving during DNA extension. So you need to know these enzymes. So this is the last part. You need to know what DNA replication is, the mechanism, how lagging strand happens, how leading strand happens. You need to know the enzymes that are involved. The enzymes that are involved. You need to take note of that. I don't know if there's any questions.